we get samples from the field, from different individuals, and we can get samples and this is very important, we can get samples from blood, we get samples from saliva, we get samples from the hair. So what we can get samples from anything that we can get DNA from, that we can get cells from, so we can get DNA. So a lot of times, most of our samples comes in a blood tube. I know everybody's been to the doctor. They actually have collected blood. So this, of course, is not in blood. And this one, we normally get samples in blood tubes. Those samples are then taken, and we collect. In this one, we spin the samples down and collect. I should have took the picture. I didn't take the picture. We collect the white blood cells with a little red blood cells mixed together. So when you spin down blood, you have plasma. You have plasma, which I didn't show on the wall. We have plasma, then you have the white blood cells, and then you have the red blood cells. So what we're interested in when we get DNA will come from the white blood cells, okay? The nuclear material from the white blood cells. So we normally get, these are a little bit of red blood cells and white blood cells mixed together. We normally take this sample, we mix it with a solution that's called red blood cell lysis, which actually just lyses the red blood cells, and it end up a little darker than this. And then with this one, just don't have a lot in there. You end up with just red blood cells and white blood cells, the red blood cells being lysed. And we spin that down, and we end up with the protein at the bottom, and the tube will have just DNA.
When you say spin it down,
I'm sorry, I forgot. What's this? I asked Marty from last year. You remember what this is? What?
It's a mixer thing that you that mix.
No, that's not the mixer thing.
Oh.
This is the mixer thing.
Oh.
This is the vortex that we mixed with. You remember the vortex from last year? Mm-hmm. We mixed up with the vortex. We actually did the experiment last year. We mixed with the vortex. Then with the vortex, like we did that, we spun it down in a what?
In a tube?
Tube in a, you remember?
Centrifuge.
So what happens with this, and I only have a small amount of DNA in here, and if you all want to get a glove, these are big, so everybody can use these. And if you can't see it that well, and I had more DNA, the DNA will come out in a little bitty white material. Can you see it? It's spinning around.
Oh yeah, I see it.
And so if you see it, this is a small one. You normally have something
Yeah, I
about 10
see.
times that big. And so the DNA comes out. I'll get you a bigger picture later. The DNA comes out of solution, and we take this and we put it back into solution so we can use it in our test. So I'm sorry, guys. This is the big as it came today. I used to use 2 samples, but I didn't. Normally,
I see it.
Normally my coworker's DNA is a lot bigger. And I said, oh no,
It's a little bit.
it's just a little bit, but they can be real big. And I just wish I had done 4 or 5 different ones, but I didn't. So that DNA, we take that DNA out and put it back in solution with a buffer.
What's a buffer?
Buffer is just buffer is a solution that has all the right salts, magnesium, all the right ions and salts in it to make sure the DNA don't break down and degrade. So in that buffer, we put it back in solution so it'll be clear. It'll be clear like this. It'll be clear. You won't be able to see it again. It looks just like water.
How can you tell whose DNA is it?
So what we do, that's a good question. So like on this sample, it has an ID that we scan in. This particular tube has a 1D ID and a 2D ID. So we have to have a 2D reader to read this one. We could have just a plain old scanner to read this one. So we have scanners that read 1D, and this one is a 2D. So we give them two different IDs, and that ID follows this sample all the way through. So this ID comes with us from the field center. And when we get ready to extract, we use the same ID in all the tubes that we have. So all the tubes will have the matching ID when we actually do our experiment. So all of these DNA will have the same number. And then when we come to the end of our experiment, the DNA will be in another tube like this. It'll be clear. And we take this sample, and you will see when you go see Genesis, how we take the sample out of here, on the robot, we had to scan everything in on the robot. On the robot, we had to scan in everything on the robot, and the robot knows to go pick this up and put it in in a certain location. So then we track everything. We scan everything in when we come in. We manually scan again. We triple check when we actually doing extractions. And then we end up taking the sample out of this tube, putting it into another tube that doesn't have that has a 2D barcode only on it. And then the robot tracks that, and our informatics tracks that from now on until probably forever. So after we get these samples, we actually look at the quality of the DNA. And I should have brought you a picture. And we actually run a gel. And this is called, and this is called gel electrophoresis. I'll just take one out. This is a used one. So we take the sample, the DNA sample, and we mix it with a dye, and we actually can run, I should load a gel for you. Let me load one. I had to restock this lab. They just cleaned this lab up. And I'm just gonna load a gel. And so y'all know, in the lab, this is called gel electrophoresis. This is called a plate. So when somebody say in the lab they're talking about a plate, it's just what they put in their sample in. Like your plate at home, you put your food on the plate. So in the lab, we say, where's the plate? Where's my DNA plate? Where's my RNA plate? Or where's my... So this is called a pipetter. It actually pipettes stuff up and down. This is called a pipette tip. And you learn all this. Have everybody had this in school yet?
What?
Working with pipettes and pipette tips? Not yet. You all should have it in high school for sure. And the purpose of this is just to, I forgot, I have a lot to say and I don't have that much time. The purpose of this is to just put a certain amount of volume into your experiment. And this particular machine holds about, y'all know about microliters? That's a math question. I had math today. Y'all know about microliters, milliliters, liters? Yes.
Yeah,
Okay.
milliliters.
Liters and milliliters and microliters, and it's nanoliters and deciliters.
No.
So we know to work with microliters in our experiment. I know y'all had that already. And you just take the sample and stick it in. This takes practice. You stick it, there's a little well there. If y'all want to look, there's a little well. You see that little well there?
Oh, yeah.
And you just take it and you make sure you put the sample in that little well. And you put it in that little well, it actually will run when I cut the voltage on it. We take those samples and we actually put them into another container that we store in the freezer. So we take all those samples and we'll make a decision as to what we're going to do with the samples. Are we just going to store for future use, or are we going to use it right away? So a lot of our samples are stored in racks like these. And it's another container. We store samples in there. Everybody has a 2D barcode at the bottom. So I know what person that is based on their 2D barcode. And that information is where Genesis come in. He actually takes all the samples that comes into the field center. It comes into us. We put it on the computer-based programs we have where we scan everything in, and that sample is then tracked from the time it comes into the door until we actually use it on an experiment.